Hello everybody, welcome back to Microbiology. I'm Mary Annarella, your host, and we are getting into Chapter 8 right now. First up, you will be happy to know that I appear to have fixed the problem with my Apple Pencil. I had to disconnect it from the iPad, reconnect it, and that seemed to have fixed the problem. So, it's working again. And we're moving on to, we have been talking about lately about how mutations occur in bacterial cells, actually, and in eukaryotic cells as well. They can happen randomly, and you can get, where'd we go? You can get base substitutions, point mutations. You know, if DNA polymerase makes a mistake and it doesn't proofread it properly, you can get a missense or a nonsense mutation. Adding or removing a base causes a frame shift mutation. Transposable elements occur in a lot of different organisms where genes will essentially excise themselves from their location on a chromosome and reinsert themselves. Mutations can be induced through chemicals. It can also be induced through through transposons or irradiation, such as with X-rays and UV light. This is the reason that we wear sunscreen. We wear sunscreen to prevent UV light from penetrating into our skin cells and damaging our DNA, causing thymine dimers. And this is also the reason that you got to wear those lead aprons when you have an X-ray done. It's to prevent the X-rays from damaging your DNA and causing a possible mutation. Of course, they can be repaired, and we talked about mismatching repair. We talked about repairing thymine dimers. But let's say some mutation occurs in a... Oh, you know what? I did want to also mention something about irradiation. Let's go back here for a sec. X-ray irradiation is often used in plants to deliberately cause a mutation, such as to give desirable new traits that we want. Otherwise, crossbreeding plants takes a very, very long time, and it's difficult. You just kind of have to get lucky to find, a tomato that makes a protein that lets its flavor last longer. If you put it in the fridge, it won't lose its flavor. Ruby red grapefruits, for example, were developed through the irradiation process. Regular grapefruits were irradiated, and these ruby reds were the actual result. So, all the talk about genetically modified foods. Texas rubies are, not just Texas rubies, but ruby red grapefruits are genetically modified organisms. They aren't genetically modified. They were genetically modified on purpose by exposing them to irradiation. And we ended up with a trait that people ended up liking the flavor. It's a slightly different flavor. So inducing a mutation is often used with a lot of plants, for example, to get traits that we find desirable, that we want. For example, a rice that will grow in the desert. That would be helpful. Or rice, like golden rice. Golden rice is a different mechanism. But for example, certain foods that have more nutrition, so you can take them to food deserts, and people can get more nutrition out of a particular type of food. So it's all about acquiring new traits, and this happens in bacteria, obviously. Like, let's say a mutation occurs, where you get a mutation. I'm gonna make this a little thinner so you can see this better. A mutation occurs in, say, a bacterial cell, and there's a new trait. When this cell divides into two daughter cells, you're going to have two cells with that newly acquired trait. Okay, passing traits on to daughter cells is called vertical gene transfer. But sometimes you can get gene transfer from one cell that, let's say, has some newly acquired trait on its chromosome, and there will be some whole different bacterial cell over here that doesn't have the trait, and this gene can be transferred from one cell to another. This is not passing things on to daughter cells, so this is actually horizontal gene transfer. Horizontal gene transfer. It's transfer of DNA from one cell to another cell. If these are genetically different species, it's called genetic recombination. So let's take a look at the PowerPoint for this. Come back. Here we go. Horizontal gene transfer, addition of DNA from one cell to another. There are 3 different mechanisms that we will talk about there. Genetic recombination, it's horizontal gene transfer when the 2 cells are genetically different species. So how do we do this? Genetic recombination, horizontal gene transfer, because it happens a lot between prokaryotic cells. Three major ones that we will talk about. DNA-mediated transformation. I have here that you did this in the lab. When we have lab in person, we would have done that experiment by now, but we have not, so you can ignore that little part. The process of transduction, which involves a virus, and then the process of conjugation. We already talked about transposition, so we're just going to focus on these first 3. First up is DNA-mediated transformation. It's when naked DNA is taken into a cell, incorporated into a bacterial cell's genome. So let's talk about DNA-mediated transformation, or just transformation. Transformation, and we do this in the laboratory a lot, and it does happen naturally. So naked DNA is taken into naked DNA means DNA with no cell attached to it. With no cell along with it, let's say. And it happens when a cell dies. For example, let's say we have this bacterial cell here. And when the cell dies, its cell membrane and all the cellular parts are going to get all chopped up. And how am I going to do this? Aha. The DNA will get chopped up too. And there, as you're probably aware, there is DNA all over our environment. Okay, we leave little bits of our chopped up DNA from our dead skin cells on everything we touch. That's part of the reason that DNA fingerprinting actually works, because we can swab DNA from a doorknob or from a crime scene, for example, and try to match these bits of DNA. So let's say, however, for all intents and purposes, that there is this little piece of DNA here that is left over from a bacterial cell. And let's say that on this little piece of DNA here, there is a gene for streptomycin resistance, for some sort of antibiotic resistance. Let's say streptomycin resistance. Okay, this little thing that I'm putting in here, this stands for streptomycin. It's an antibiotic, and that capital R stands for resistance. So any bacterial cell that has, I've got to use the white again, any bacterial cell that has the gene for streptomycin resistance is going to be able to grow in the presence of streptomycin. It's not going to die. It's going to be resistant to it. So streptomycin won't kill a bacterial cell. So keeping in mind that this is naked DNA now from some now dead cell. Well, let's say that there is another cell in the environment. It's just hanging out, minding its own business. If it touches this little piece of DNA that came from another cell, there, I'll just draw it like that. Under certain conditions, this piece of DNA, and you know what, I'm going to draw it like this now. Little double-stranded piece of DNA. under certain conditions, the cell will be able to take up this piece of DNA and basically let one strand in. So a cell that has the ability to take in a piece of naked DNA is called a competent cell. Oops, I don't know how to spell. Competent cell. It's a cell with the ability that is able to take in a new piece of DNA. It's able to take it in. And actually we can do that in the laboratory by adding calcium chloride salt to it, for example. But only one strand enters the cell. Not both. One strand enters the cell. What can happen then is that when that one strand enters the cell, it can find a region of homology, meaning a region that has a similar sequence, a similar order of G's, A's, T's, and C's. So one strand enters the cell, and if it finds, I don't want to anthropomorphize this. I don't want to say that the DNA has a mind of its own, but if there is a location of an area of homology, through a process called homologous recombination, it can actually get itself inserted into the genome of this new cell. It can insert, it doesn't really insert itself. It gets added to. That would be a better way to say this. It can become integrated into the new cell's chromosome by a process called homologous recombination. It's a mouthful, I know. Homologous recombination. So check this out. This blue cell here just took in DNA from this cell, from a long ago dead cell. Remember, there was a cell here? Remember? The cell is all gone now. Cell is gone. Bye-bye. All right. But this red cell that is now dead and this blue cell that is still alive now has a piece of the dead cell's DNA in there. These cells never had to touch each other. And they never saw each other. They never knew each other. They never said hello. Nothing. But if this has a gene for streptomycin resistance on it, this cell has now acquired that new trait. So this is one way that cells can pass on new traits, such as antibiotic resistance. Really interesting stuff. Now there is another way that this can happen, that horizontal gene transfer can happen, and it's through the process, or what are we doing, A, B, C, D here? The process of not transformation, but transduction. And this involves a virus. Now here's something about viruses. When viruses are host specific, generally, until they learn, like coronavirus mutated so that it could not only infect pig cells, but it could infect human cells. But for the most part, viruses are fairly host specific until they acquire some sort of mutation that will allow them to jump the species barrier. So virus, and in general, because bacterial cells are so different from human cells, viruses that infect bacteria generally only infect bacteria, and they're never going to mutate enough to the point where they can infect humans, right? But a virus that infects bacteria, we actually have a specific name for them. They are called phage, or sometimes they're called phage. I don't really know which pronunciation is correct because I hear both in the scientific world. So phage, or sometimes we use the Greek letter psi for that. It's a Greek letter there. So phage or bacteriophage or phage, bacteriophage. Maybe if you speak it with a mid-Atlantic accent, I don't know. Or maybe Moira on the show of Schitt's Creek would call it bacteriophage. Phage. Jocelyn, that's a phage. I don't think Moira would ever talk science. If you know the show, it's called this. That's what I'm referring to. I didn't just swear. It's a different spelling, Schitt's Creek. One of my favorite shows, and Moira has a very odd accent. So I don't know. Maybe Moira would say bacteriophage instead of bacteriophage, or just phage. Now, if you remember, viruses and phage or viruses, it's just a different word that we use. Like if we say the word phage, we know we're talking about viruses that only infect bacteria. You know that viruses have, let's go with a different color for viruses. Let's go with, I don't know, let's go with purple. They have a little piece of DNA or RNA. That's their genome. And they'll have a little protein coat around them. Here we go. And these protein coats can take on various shapes. And there is this one bacteriophage that's called T4 bacteriophage, and it's got this funky looking protein coat that looks like this. I mean, it looks like a lunar module, and then it has all these protein spikes on it right there. Like this is T4 bacteriophage right here. So there's its DNA. All right, when this guy infects bacteria, let me use, no, that doesn't, use green here. Here's a green bacterial cell right here. Well, this little phage or phage will basically land right on the cell, and it will attach, kind of like coronavirus will attach to human cells through the ACE2 receptors there. And what it does is it puts a tiny little nick in the cell membrane, and only its genome gets in. Only its little piece of DNA gets in. It's actually called uncoating. It's the uncoating process because the protein coat stays on the outside. So we're going to have this empty protein coat. There we go. So this stays on the outside. I'm going to erase all the purple stuff. You stay out there. But its DNA just got inside. When T4 bacteriophage, it's called the lytic cycle, gets its DNA inside a cell, it basically takes over all the other cellular machinery, like the ribosomes and everything, and its DNA polymerase, and it actually will chop up the host cell DNA into little bits. Oh, no, that's not where I was going with this. Actually, it does do that eventually. Crap, my bad. Let's back up just a little bit because I don't feel like starting a whole new lecture screen capture right now. Let me put this genome back together. There we go. This little guy, through genetic recombination, homologous recombination, gets its genome inserted into the host cell's genome. Then it takes over, it gets its DNA copied. Oh, wait, I'm going to do that. Do this here. It gets copies of its DNA made. Then it chops, oh shoot, that did not work the way I wanted it to. Then it chops up, shoot, I did not want to do that. Then it chops up the host cell DNA. Sorry, I got a little ahead of myself there. All right, because what it's trying to do is take over everything in the cell and say, ha, you are not getting any of your host genes made. We're going to make only my viral genes and I will take over the cell. So eventually it does that. But, oh, come back here. I guess I didn't want to do quite that. Sometimes little bits of the host cell DNA remains. So as it tricks the cell into making its little protein coat, these little lunar modules here, it basically turns the cell into just a factory for making viral particles. Like that. And it gets its DNA all packaged up inside. But sometimes, imagine if this one went in here, a little bit of the green DNA that came from the host DNA is going to be in there, in here. So when enough of these viral particles are made inside the cell, it actually produces the virus produces some proteins that will actually break apart the cell membrane. So boom, it's gone. And then these guys will all go on to infect new bacterial cells. Well, let's say that this one right here goes on to infect yet another bacterial cell. And let's do this second bacterial cell in red. And this one lands on here, gets its viral DNA in there, but that viral DNA has some of the DNA from that previous cell. And what if that gets integrated into the new bacterial cell's genome? This green DNA here came from another bacterial cell, but it was merely carried there from the old cell to the new cell. So in this case, transduction, a virus carried a teeny bit, these little green bits of bacterial DNA from the previous cell that it infected. So a virus is carrying DNA from one cell to another. And you know what? This red cell never met the green cell before. They never had to touch each other at all. We will spend a little more time talking about viruses and how they infect cells. I know you'll have some questions about this, and we will address that in another chapter. Totally fine. You'll be held responsible for more of the details of how, the chopping up of the host cell genome occurs and that later. But for now, just know that transduction, it's where a virus that infects bacteria, bacteriophage, carries DNA from one cell that it's infected to the next cell that it's going to infect. And those two cells never had to see each other at all. Now, there's a third means of horizontal gene transfer, and it's really an important mechanism in how bacteria actually share genes with each other, and that is called conjugation. It's sex for bacteria. It's as sexy as bacteria get. And it involves a plasmid. And remember, a plasmid is a small circular piece of DNA. But this plasmid is special. It involves a plasmid called the F plasmid. The F plasmid. The F plasmid is a plasmid, duh, with a gene on it, and I'll just draw the gene like a little box. I'll write F plasmid in the middle. And this gene contains information, codons, for building the F pilus. Now, a pilus, remember, a pilus is a bacterial cell with, okay, if a flagella is like a really long piece of protein, a pilus is shorter. The F pilus is probably the longest of all the pili, but it's an outward extension from the bacterial cell, like that. But the F pilus, so F plasmid contains the gene for the F pilus, which is also called the fertility pilus. The F pilus, actually, if we've got a bacterial cell here and it's got the F plasmid and it's able to make the F pilus, the F pilus is a very special pilus. It will actually allow connection between two bacterial cells. I'm going to draw them in different colors. Here we go. Right, there we go. The F pilus will actually allow a connection between this green cell on the left and the blue cell on the right. And it basically forms a bridge through which one strand of the F plasmid can travel. One strand of the F plasmid can travel or transfer through the F pilus. By the way, once a plasmid is inside a bacterial cell, the bacterial cell will make lots of copies of it. So it's not like this cell is not losing its plasmid. Even so, it's still keeping a strand of it. But it's got a bunch of them. So now you have two cells that contain the F plasmid and can make that pilus. Now here's the deal with the F plasmid. Sure, it has the gene for the F pilus on it, but it also, because there are a lot of different F plasmids out there, I don't want to fool you into thinking there's only one type of F plasmid and that's it. They often have genes for antibiotic resistance. Oh, I can't spell resistance. Looks like restance. Resistance gene. So, if this antibiotic resistance gene is a penicillin resistance gene, penicillin resistance, I'll write it like that, now we've got two cells with penicillin resistance. Plasmids can also recombine when they're inside the cell, and if new traits are acquired there, this cell can make the F plasmid and pass that plasmid on to yet another cell, right? You can get whole huge populations of cells that end up with antibiotic resistance due to conjugation. So conjugation is a major route through which bacterial cells share the plasmids with one another and create more cells that have antibiotic resistance, and it is a huge problem. We actually do a conjugation experiment in the laboratory where we will select for this cell to make sure that conjugation actually occurred, all right? And since we're not in person, we are doing a simulation in laboratory. So we have a conjugation experiment in lab. I know we have a Connect one, but no, this is actually not a Connect one. This is actually a PDF that you'll be sent probably next week from most people's labs. I know my lab's going to be doing it next week, and I think Rubinsky's lab will be doing it next week. But it's a PDF with the procedure and results. And it's going to be, we'll explain it in lab a little bit more, but we'll give you pictures of the results that you see, and you need to write up what's happening. But the point is that with conjugation, these cells, they have to meet each other, right? They got to get together to conjugate. When we mix them together in the laboratory, one of the now retired microbiology professors is like, okay, we're going to turn down the lights, play a little Barry White music for our bacteria. And all the students just looked at him like, what? It's a microbiology joke. This is the best sex that bacteria get. It's as sexy as they get. They get to conjugate. They get to connect to each other by a plasmid. And just to kind of demonstrate what I mean, going back to the PowerPoint that I have here for you, this is kind of hilarious. If I come down here, here's an actual micrograph of the F pilus, also known as the Sex Pilus, not to be confused with the Sex Pistols, the band, punk band. 80s, never mind. But anyhow, it's a very large one. So these two bacterial cells are transferring DNA between each other. All right, so this is the only type of horizontal gene transfer where the two cells have to have contact with each other. Oh, and don't worry about this stuff about F-. F- cell means that it doesn't have the F plasmid. F plus simply means that they both have it then. So that's all that means. But here we go. We're just, oh my God. I stopped showing this to my classes, but I left it in the PowerPoint just so that you know what's going on here. But here we go. Here's the donor cell right there with the F plasmid. Here's the cell that's F-. It does not have the plasmid. This one makes the pilus. And yes, this looks so phallic. It is absolutely ridiculous. My students were all just laughing. I'm like, what? Stop. They're like, it looks ridiculous. But they don't, that's not accurate. They don't fuse to each other like that. It's more like there's a bridge here, and that's about all. But we do know that one strand of the plasmid enters. And then both cells have the plasmid right there. So there we go. Delete that. I'm not going to worry about this HFR cell thing that's down here at the bottom of this slide here. You get a high frequency of replication there. Don't worry about that. I'm not gonna worry about it. You can scratch out this slide too. I don't care about that. And yeah, these last three slides in the PowerPoint, don't worry about them. This one, and this. Oh, well, whoa, there actually is one more. Yeah, this one, keep this one. This one, go ahead and cross that out because that's got transposable elements there for transposon. Cross this one out. Cross this one out, and we're good there. Yeah, but I do want to talk about plasmids. So this last slide here, circle this. We want to save it because thinking back to DNA-mediated transformation, that's what I want to talk about next because we do this in the laboratory. So back to transformation. That's where naked DNA is introduced, I can't spell, into a living cell. And way back here, I was like, okay, here's this piece of DNA from a long ago dead cell, like right there. We engineer these pieces of DNA in the laboratory now in plasmid form because it turns out that bacteria really like to take in plasmids. So we'll have a plasmid here. And what we can do is we'll have something that causes ampicillin resistance. And I'll just put an AMP-R. It's an ampicillin resistance gene on it. But also there's a little spot here called a multiple cloning site. Multiple cloning site, where we can use something called restriction enzymes to insert our favorite gene, or a gene that we want to study, or maybe a piece of DNA that we want to make more of. So into the multiple cloning site, or the MCS, we can insert, in the laboratory, we can insert other genes. Like maybe there's some protein that we want to make more of, and it's easy to trick E. coli into making it. So I'm going to put in a different color for that gene of no, that's not a stark enough contrast there. Here we go. So here, this little black piece in there, that's our gene X. We can, in the laboratory, have an E. coli cell, and we can introduce our plasmid and get it to take up that piece of DNA with our gene X. Now, this bacterial cell, this E. coli cell, is going to be resistant to ampicillin, and that's actually how we can make sure that the plasmid got in there. If we grow this in the presence of ampicillin, it's going to be able to grow in the presence of ampicillin. That means that it's got the plasmid in there, and we hope also that it will make our gene X product. So we use that in the laboratory a lot. It's one way that we genetically this is a form of genetic engineering. It's one form of genetic engineering. It's one way that we get bacteria to make certain proteins that we would like to have lots of, but are harder to get lots of in other ways. So it's one way that we've genetically engineered bacteria to do that for us. And that is plenty for today. When we come back, we'll talk more about viruses, and we'll start moving on into that direction. So have a great day, and we'll talk in class. Bye-bye.